Right, in this video, we're going to look at dihybrid crosses and how Mendel's experiments led to the development of the law of independent assortment. Now, there are two big questions that Mendel was asking. Number one is, do the two characters yield the same results as in a monohybrid cross? Another corollary of that is, are these characters linked to each other? In other words, if you have round seeds, are they always yellow, or if they are wrinkled, are they always green? Now, Mendel was working with dihybrid crosses. A dihybrid cross means that you're crossing individuals that differ in two characters. An example of that would be like seed texture versus seed color. And then these are the traits for these characters. Now, there's two possibilities. Or two possible patterns of inheritance here. One is linked assortment, and that's on this side of the diagram or the figure. The second hypothesis could be independent assortment, which is diagrammed on the right side of this figure. So let's first talk about linked assortment and what it would look like in these subsequent generations. Now again, what we're starting out with is our P generation. These are true breeders, meaning that they're homozygous for both of these for both of the traits for these characters. So we've got round and yellow here, and then wrinkled and green here. So what you would expect to see the produced from these individuals, the haploid gametes, from an individual that only has the big R allele for the round seed texture, or the big Y allele for the yellow seed color, they can only produce big R, big Y. And that same kind of reasoning goes here, in that the only type of gametes, haploid gametes that this individual could produce would be little r, little y. So what's happening here then is that every time you have a round seed, you've also got a yellow round texture, you've got a yellow color. So in the F1, we still would get heterozygotes. Those individuals would express the dominant traits, that would be round and yellow. Again, because these are linked, we would only produce gametes that have the big R allele and the big Y allele together, and gametes that the other half of the gametes would only be the ones with the little r and the little y allele. So for what I want you to think about here is what the F2 would look like when you mate these individuals. Now, in terms of independent assortment, we're starting out at the same place with this P generation that are true breeders. So these are the only possible gametes that these individuals could produce. This individual can only produce R and Y, and this one big R, big Y, and little r and little y. Again, we get to our F1, we get these all heterozygotes. This is typical from what we've seen before with our monohybrid crosses. Now, at this point, under this idea of independent assortment, there's two other, if you will, or two more possible combinations of these alleles, big R, big Y, and little y, that you can have in your gametes. And there's an equal probability that you can produce each of these individual gametes because they're assorting independently of each other. So you could have big R and big Y. You could also have big R and little y. You could have little r and big Y, or finally little r and little y. So these two right here, these two gametes, represent different varieties, if you will, of gametes than what we would see under the hypothesis of linked assortment. So let's talk about that a little bit more. For this one, under linked assortment, essentially we're doing a monohybrid cross, because the only gametes that can be produced here for one of the individuals would be big R, big Y, and little r, little y. Sorry, that's a little y. Now, remember, these represent the haploid gametes. We can do a simple Punnett square with this. So over here, with another individual, and these genes are linked, and therefore their alleles are linked, these would be the only possible gametes also produced. So you'll notice that every time we're going to get these gametes, or I'm sorry, these alleles arranged in this manner. So we've got R little y and R little y finally. So that the first one here represents this gamete. This one here is representing the gametes in the second along down the column. So there's only two types of gametes that can be produced at any of these generations. Your offspring are always going to be in this kind of arrangement, i.e. this is a 1 to 2 to 1 genotypic ratio, and this would be a 3 to 1 phenotypic ratio. Now let's look at it under the situation of independent assortment. From your, if you look down here at the types of gametes that can be produced, we know that one individual can produce these 4 different types of gametes. Those would go along the top of our Punnett square. So that's essentially R big Y, R little y, big R little y, and little r and little y. Now we would do the same thing for the second parent or the second gamete, going down that should be a big Y and then a little y. So now we've got 4 possible gametes produced by each parent, and then, so that's 4 gametes, 4 types of gametes. And that is, there's an equation where you can actually figure out if you have 2 heterozygous conditions for 2 traits, that is 2 to the n, where n equals the number of heterozygous conditions in that individual. So here we've got 2, so that's 2 to the second, which equals 4 possible gametes. So that's a way to check to make sure you've got all the possible combinations of those 4 different alleles in the gametes produced from one individual. Now what you'll also notice is that if you have 4 gametes from one individual and we're mating it against a double heterozygote again, 4 gametes for another individual, that means you got 16 possible combinations of these 4 gametes. Now let's look at the data from the crosses. So here's the data for just the round and yellow seeds crossed with wrinkled and green seeds. Remember again, these are true breeders, so that means they're homozygous for both of those characters. They have both of the same alleles for them. In the F1, we get our all heterozygotes, which that's typical of what we've seen before, because we're only seeing all round and all yellow. And I should step back for a second and say, and that's the parental types that we see in terms of at least the dominant trait. Now when we get to our F2 generation, we start to see something different. You remember that in the F2 generation for the monohybrid crosses, we typically only saw 2 parentals, i.e. we saw the reappearance of that recessive trait in of those recessive traits in the F2 generation. That represents this parent here, which is wrinkled and green. Now these are non-parentals in the sense that we don't see that combination of traits in these parents. So let's talk about how we interpret this data. So again, we're seeing these seeds with novel combinations, not found in the parentals. We get round and green and wrinkled and yellow. The fact that we see these, that contradicts this idea of a linkage model. Because if it was linkage, then we would only see round and yellow and green and wrinkled. Yes, that does say green and wrinkled. Now, if the genes assort independently, i.e. they're not affected, they're not linked to each other, then we should expect to see a phenotypic ratio of 9 to 3 to 3 to 1 in the F2 generation. Not in the F1, but rather in the F2 generation. And what we see, based on these numbers, is that it is very, very close to a 9 to 3 to 3 to 1 ratio. Now, based on this data, Mendel proposed the law of independent assortment. So essentially what we're saying here is that during the gamete formation, the segregation of any of these pairs of genes is independent of the segregation of the other pairs. So the segregation of the alleles for the round gene, round character, are independent of the segregation of the texture of the seeds, i.e. the wrinkled, or I mean the color of the character, so the yellow and green. Now let's look at where this 9 to 3 to 3 to 1 ratio comes from, and recognize that this is a phenotypic ratio. So under this situation, or in this scenario, we've got our 4 possible gametes from a male plant, or in 4 possible gametes from the female. They're the same because both of them are double heterozygotes. These are all the possible ways these can combine, and you could go back to that Punnett square, and you can demonstrate all these possible combinations. Now, phenotypically, that's really what we're interested in here, is what is happening phenotypically. So all of these genotypes would correspond to round and yellow seeds. All of these genotypes would correspond to round and green. All of these wrinkled and yellow, and all of these would be the wrinkled and green seed. Now, finally, I want to remind you of this, that this type of you could do a dihybrid test cross, and this will come up later when we talk more about genetic linkage. You can also see if you can test for independent assortment this way. So in this situation, we're crossing one of those F2s with basically one of the true breeders. Now, the only type of alleles that the recessive homozygote can contribute are TY, because that's the only alleles that they have. However, again, because of independent assortment, we can get 4 possible haploid gametes from this parent that is a double heterozygote. If we put these together, we should see a 1 to 1 to 1 to 1 ratio in terms of those phenotypes. Now, this is only in the situation where you have a dihybrid test cross. Okay, if this individual was a double heterozygote also, would you see a 1 to 1 to 1 to 1 phenotypic ratio? No, you would see a 9 to 3 to 3 to 1. So these dihybrid test crosses are used quite frequently in trying to determine if this parent is actually a double heterozygote, and also to test for linkage.